Welcome to your rapid-fire revision guide for GCSE Biology. We're going to tackle cell structure, a really fundamental topic from the AQA spec. In just the next few minutes, we're going to cover everything you need to know to feel super confident and totally exam ready. So let's get straight into it. So let's kick things off with a big question, shall we? The answer really gets to the heart of cell biology, and it introduces the two basic types of cells we're about to explore. We'll break down their structures, see how they specialize, and then master that key formula you're going to need for your exam. All right, let's dive in. This right here is the first and honestly the most crucial distinction you have to get right, the difference between prokaryotic and eukaryotic cells. Pretty much everything else in this topic builds from this one idea. Okay, so here are the core differences, plain and simple. On the left, you've got prokaryotes, things like bacteria. They're simple, they're small, and the absolute key takeaway is they have no nucleus. Their genetic material just kind of floats around in the cytoplasm. But on the right, you have eukaryotes. That's the cell type in plants and animals like us. They're bigger, way more complex, and they keep their DNA tucked away neatly inside a nucleus.
Right, so let's zoom in a bit on those more complex eukaryotic cells. For your exam, you really need to focus on the two main types, animal cells and plant cells. We're going to look at what they have in common by comparing their subcellular structures, which, by the way, is just a fancy name for all the smaller parts inside a cell. And, really importantly, what sets them apart. As you can see, both of them share 5 key structures: the nucleus, cytoplasm, cell membrane, mitochondria, and ribosomes. They've all got those. But, and this is the important part for your exam, you need to know the 3 structures that only plant cells have. A brilliant way to remember them is with the 3 C's: a cell wall, chloroplasts, and a large central or permanent vacuole. Look, it's not enough to just name these parts. You have to link the structure to its function. Think of the nucleus as the cell's control center, and mitochondria as the tiny powerhouses releasing all the energy. Ribosomes are like little factories that build proteins. And, unique to plants, chloroplasts are where photosynthesis happens, while that cell wall gives the plant strength. Knowing these connections, well, that's where you'll pick up some really easy marks in an exam.
So let's see how this all builds up. Cells don't just stay as these generic templates, you know? They actually go through a process to become specialists, each with a very specific job to do. And this process has a name. It's called differentiation. This whole process explains how a complex organism, like you, is built from just a single starting cell. An unspecialized cell, think of it like a trainee, develops new features and different parts, and that transforms it into an expert, or what we call a specialized cell, that's perfectly adapted for its role.
Now, in your exam, you will almost certainly be asked about examples just like these. You'll need to explain how a sperm cell's tail helps it swim, or how a nerve cell's long shape helps it send signals really quickly, or why muscle cells are just jam-packed with mitochondria for the energy they need to contract. It is always about linking that structure to its function.
And you know what? The exact same idea applies to plants. A root hair cell, for example, has this huge surface area so it can absorb water and minerals super efficiently. And xylem and phloem cells, they form the plant's plumbing system, and they're specialized for moving stuff up and down the plant. But hang on a second. How do we know any of this stuff? We can't see cells with our naked eyes. The invention of the microscope was a complete and utter game changer for biology. It allowed us to finally peer into this incredible hidden world for the very first time. We started off with light microscopes, which are still used in schools, but they definitely have their limits. The key difference you need to know is that electron microscopes have a much higher magnification and a much higher resolving power. Resolving power is basically just the ability to tell 2 points apart, so a higher value means you get a much clearer, more detailed image. This massive leap in technology was revolutionary. It meant we could finally see all these tiny subcellular structures like mitochondria and ribosomes in incredible detail. And this wasn't just about seeing smaller things. It fundamentally changed our understanding of how cells actually work. Okay, finally, let's get you totally exam ready with the one formula from this topic that you absolutely must know. And don't worry, it's really straightforward once you see the steps. This is it. You have to commit this to memory. Magnification equals the size of the image divided by the size of the real object. Now, in the exam, they might ask you to use the formula just like this, or they might ask you to rearrange it to find one of the other values. Following these 3 steps is the secret to getting full marks on these questions every single time. Step 1. Always, always write the formula down. That's often worth a mark on its own. Step 2, and this is the crucial one, listen up. You must make sure your units are the same. If one is in millimeters and the other is in micrometers, you have to convert one of them before you do anything else. Only then, for step 3, do you plug the numbers in and do the calculation.
So here's a typical calculation. Let's say your image is 30 millimeters and the real object is 20 micrometers. First thing you do is convert the units. So 30 millimeters becomes 30,000 micrometers. Then you just divide 30,000 by 20, which gives you a magnification of 1,500 times. It just shows you how incredibly powerful these microscopes are. So let's quickly recap the absolute essentials. Remember, prokaryotes are simple, no nucleus. Eukaryotes are complex, know the 3 extra parts of a plant cell, the 3 C's. Understand that cells differentiate to become specialized. And finally, master that magnification formula, and please, never, ever forget to convert your unit.
Okay, I'll leave you with one final thought to kind of connect all these ideas. Most animal cells, they lose the ability to differentiate very early on. But lots of plant cells, they can do it throughout their entire lives. And that is precisely why you can take a cutting from a plant, stick it in some soil, and grow a whole new one from it. It's an amazing example of differentiation in action. Good luck with your revision.